What if I told you that you could lose weight just by listening to music? Something as simple as sound waves, aka music, can now alter your genetic activity and suppress fat cell formation. Yes, this is true, and it works. It was already tested in mouse cells, and it works. Frequencies like 440 Hz and 14 kHz were shown to alter the expression of dozens of genes. They blocked preadipocytes from maturing into fat-storing cells, thus reducing lipid accumulation by about 15%. This is all possible because of the research that is being conducted by Japanese scientist Masahiro Kumeta, who is a cell biologist at the University of Kyoto. You see, preadipocytes are immature fat cells. They can't store fat yet. They need to be activated in order to become adipocytes, which are mature fat cells that store fat. When there is need for more fat storage, that is if you take in more calories than your body needs, then your body, specifically through some mechanisms, work together to push preadipocytes, which are immature fat cells, into becoming adipocytes. In Masahiro Kumeta's research, sound waves at a certain frequency can stop these preadipocytes from becoming adipocytes. They do this by influencing proteins such as peroxisome proliferator activated receptor gamma and CCAT, or enhancer binding protein alpha. These are the protein receptors that push preadipocytes into maturing into adipocytes in order to store fat in the body. This means that there would be fewer fat cells to store fat because preadipocytes won't mature into fat-storing cells. Masahiro Kumeta has been investigating the relationship between sound and cells for more than a decade, and in 2018 there was a breakthrough. He found out together with his colleagues that audible sound waves could modulate the expression of various mechanosensitive genes in cultured cells. What are mechanosensitive genes, you may ask? Well, these are genes that respond to mechanical stimulation, such as changes in membrane tension, pressure, or shear stress. These genes play a crucial role in various biological processes by encoding proteins that act as mechanoreceptors, translating mechanical forces into biochemical signal. Sound is a mechanical force. Sound is made up of pressure waves that travel through air, water, and tissues. These waves physically interact with cells, even at low intensities like 100 pascals, similar to gentle pressure experienced inside the body. Cells have built-in sensors, that is, mechanosensitive proteins, that detect mechanical changes like stretch, vibration, or pressure. These sensors are part of the cytoskeleton, which connects the cell's outer surface to its nucleus and gene control mechanisms. Different frequencies affect cells differently. In his research, single sound frequencies were used, specifically 14 kHz and 440 Hz, O and white noise. They were produced using NCH tone generator software. And the sound intensity was recorded directly using a hydrophone in water and the pressure level calculated. Single frequencies like 14 kHz and 440 Hz were selected as representatives of low and high audible frequencies, respectively, while white noise was chosen as a broadband source with uniform frequency characteristics. In the first 2 hours, similar gene activities were seen across all sound types, and 42 genes were expressed. These genes were mostly involved in cell adhesion, migration, and cytoskeleton remodeling. After 24 hours, this was when the differences between frequencies became more distinct. 14 kHz, which is a high frequency, upregulated certain genes. It turned them on. 440 Hz, a low frequency, downregulated those same genes. So what does this mean? 440 Hz creates larger particle displacement and more fluid movement, like stirring water. 14 kHz produces smaller vibrations, with less fluid motion but deeper pressure penetration. The 14 kHz sound created less fluid motion, which in the culture medium reduced oxygen circulation. As a result, hypoxia-related genes, which are genes activated in low oxygen environments, were turned on at 14 kHz. So 440 Hz, which is a low frequency, created a stronger fluid motion, which equal greater mechanical stress. This mechanical stress affects the cytoskeleton of the cells, thus leading to their inactivation. Hence, it suppressed fat-related genes. 14 kHz, which is a higher frequency, created less fluid motion, thus mild hypoxia. It activated different gene pathways, possibly mimicking low oxygen stress. White noise had a broad band response. It triggered both types of reactions, but weaker than specific frequencies. Some genes were upregulated by 14 kHz and downregulated by 440 Hz. I know you might be confused, so let us try to dissect this slowly. Low frequency sound mechanically stimulates focal adhesion kinase, which is an enzyme at the cell membrane. It rattles the cell cytoskeleton and sends an alarm via focal adhesion kinase to turn off the genes such as SEPA and PARG. FAK phosphorylation increases, thus triggering PTGS2 gene expression. PTGS2 increases prostaglandin production. PGE2 binds to the EP4 receptor, which is known to block fat cell differentiation. This inhibits the fat-forming genes such as SEPA and PARG. The result is fewer cells mature into fat cells, and lipid accumulation decreases by 13 to 15%. So, the suppression of fat genes is indirect. As for the high frequency, 14 kHz has a much shorter wavelength and smaller particle displacement. So, it doesn't shake the fluid or physically jostle the cell as much, but it still exerts pressure in a more subtle, oscillating way. It is able to change the microenvironment. You see, it mixes less in the petri dish, thus mimicking a less oxygen state near the cells. As a result, cells activate hypoxia-responsive genes. These are genes that turn on when oxygen is low. They include genes tied to stress adaptation, metabolism, and cell survival. So, 14 kHz is less mechanical but more chemical in its effect. It influences intracellular signaling by changing the environment around the cell rather than pulling on the cytoskeleton. This environmental change is the one that influenced the cell's behavior. So, in this study, cells were sitting in a liquid culture medium, like a nutrient soup. For cells to survive and function, they need oxygen, nutrients, and even distribution of molecules in the fluid. Normally, fluid motion, from stirring or low-frequency sound, helps mix this soup, keeping everything evenly distributed. But because 14 kHz causes smaller fluid motion, oxygen and nutrients don't get passed around the cells that much. So, what do? The cells become super sensitive to these changes in oxygen levels, and that is when the hypoxia-related genes come in. They conserve energy and adapt metabolism to survive the stress. This lowers or puts a pause on all energy-expensive activities and focus on survival. However, in the study, there was no strong suppression of fat-related genes under 14 kHz. That effect was much clearer with 440 Hz. The most notable gene to be upregulated by the 14 kHz frequency was the DIT4. It suppressed the mTOR pathway. which controls growth and fat cell formation. By turning this on, the cell slows down growth, including fat cell differentiation. Meanwhile, 440 Hz upregulated PTIX2, which produces PGE2 that suppresses or inhibits fat cell differentiation. SEPA and PARG were turned off by 440 Hz frequency. These prevented preadipocytes from maturing. So yes, sound waves can influence fat cells, but we are going to have to wait and cross our fingers, as this research is still in the lab stages,